Welcome friends, how are you? In this video we are going to start a new topic, that is the glycosylation. I would ask you a question, what are glycoproteins? The proteins which are attached to glyco. Glyco means sugars. So the proteins which are attached to sugars or the carbohydrates, they are known as glycoproteins, and how they are formed through the process, that is glycosylation. Okay, so the glycosylation is the enzymatic addition of carbohydrates or the sugars to the proteins. Okay, now this glycosylation is of two types: N-linked glycosylation and O-linked glycosylation, and we have to differentiate between these two. The difference, as the name implies, lies where the carbohydrate or the sugar is attached to the protein. Okay, we know the protein is made up of amino acids. Okay, so when the protein which is destined for ER, it has a consensus sequence of asparagine, X, threonine and serine. This X is any amino acid except proline. So if the protein, if the carbohydrate is attaching itself to the N atom or the nitrogen atom of the amino group of asparagine, then it is referred to as N-linked glycosylation. On the other hand, if the carbohydrate is attached to the O atom of the hydroxyl group containing amino acids, that is the threonine and serine, this is called as O-linked glycosylation. This is the first major difference. Second is the timing, that is when these processes occur. The N-linked glycosylation is co-translational. That is, it happens during the protein synthesis, that is when the protein is being synthesized. On the other hand, this O-linked glycosylation is strictly post-translational. Post-translational means when the protein is fully synthesized, then we will see this process to happen. In the previous video, I said that this glycosylation is post-translation. I forgot to add that this happens in both ways, that is the co-translation and post-translation. The next difference is where does it occur, the location of this process. N-linked is in ER and its modification is in ER plus Golgi. Okay, it happens in ER but it is modified in ER plus Golgi. On the other hand, this O-linked glycosylation, this is only done in Golgi and it is also modified in Golgi. There is one more difference, and that is the length of the carbohydrate chain, oligosaccharide chain. In the N-linked glycosylation, the sugar chain is composed of 10 to 12 sugar residues. So this is larger and it is complex. On this O-linked glycosylation, this is containing short chain of oligosaccharide, 1 to 4 residues. So we have to know how this process of glycosylation occur, or in other words, how the proteins are attached to carbohydrates. But before that, there is a need to know how these carbohydrates are synthesized, where they are synthesized, and how they are assembled on each other before they are transferred to protein. So there is a preformed, that is already formed oligosaccharide chain. Oligosaccharide, oligo, many sugar units, they are attached to each other. So preformed oligosaccharide chain which is present in the ER. Okay, and this is composed of, I am going to write the residues which this this chain is containing. 3 glucose residues, and then we have 9 mannose, then we have 2 N-acetyl glucosamine or N-acetyl galactosamine residues, glucosa or galactosamine, N-acetyl glucosamine residues. And this chain, okay, this chain is attached to a system of isoprene units, actually multi-isoprene units, which is called as Dolichol phosphate. So what I just said, there is a preformed oligosaccharide, okay, which is this one, and this is attached to Dolichol phosphate, which is a system of isoprene units, and this Dolichol phosphate is present in the ER membrane. Actually, this Dolichol phosphate serves as an anchor for this chain of oligosaccharide, because these sugars, they cannot be present, they cannot be simply put in the ER on their own, so they need an anchor, and that anchor is Dolichol phosphate. The sugars are added to the Dolichol phosphate one by one before they are actually transferred to the protein. Now, before we discuss the transfer of these sugars to the Dolichol phosphate, I want to tell you some specific points regarding this mechanism. The first one is type of sugar which we are using. This is not a simple sugar, this is activated sugar. What is an activated sugar? The one which is attached to a nucleotide. So we would see 2 nucleotides, that is, one is UDP and another one is GDP, and they will be attached to different sugars. In case of UDP, this is mainly glucose, and we have N-acetyl glucosamine, second one is N-acetyl galactosamine. This GDP is attached to mainly mannose, and we have fucose. The second point we have to know is where the sugars are synthesized. All these sugars, they are formed in cytoplasm. Okay, the sugars which we would transfer to the Dolichol phosphate and then to the protein, they are all formed in cytoplasm. The third point we have is the transfer of these sugars to the Dolichol phosphate is a 2-portion process. So that means half of the sugars, they are added to the Dolichol phosphate on the cytosolic phase of the ER. Okay, and other half of the sugars, they are added to the Dolichol phosphate when it is present on the luminal phase of the ER. So now let's discuss the mechanism. So the first sugar which we attach to the Dolichol phosphate is galactosamine or glucosamine, N-acetyl glucosamine or galactosamine. This is the first sugar which is attached to UDP, this red color showing the sugar, and it is attached here to the Dolichol phosphate. What you have to note here is that in this first step 1 UMP is released. So we are using 1 phosphate from this UDP and it is attached to Dolichol phosphate. So what we are getting here? A pyrophosphate bond. So the first bond which is formed between the Dolichol phosphate and the sugar is a pyrophosphate bond, a high energy rich bond. And the bonds which would formed after this are between two sugars, and the bond between two sugars is a glycosidic bond. This is an important thing. In the second step, another molecule of N-acetyl glucosamine or galactosamine is added, and you can see here no more phosphate is used, only this pyrophosphate linkage is there. Okay, UDP is released and we have another molecule of galactosamine or N-acetyl glucosamine is added in this red color. Then is the turn of mannose. I am adding here 5 mannose residues. Actually this is one by one. For time saving I am doing this. So 5 residues of this blue color which is showing the mannose residue, they are added on this already present N-acetyl glucosamine or N-acetyl galactosamine on the Dolichol phosphate. The transfer of the sugars on the cytosolic phase of the ER to the Dolichol phosphate is done here till third step. Now it is the turn of the transfer of the sugars to the Dolichol phosphate on the luminal phase of the ER. So what we would do, we flip this. Here we flip Dolichol phosphate. Now this phosphate is facing the luminal side. But the thing here is that initially I told you that all the carbohydrates, they are synthesized or formed in this cytoplasm, but we want them on the luminal phase of the ER. Okay, so what we do here, we take another molecule of Dolichol phosphate which would act as a carrier. So here we have another Dolichol phosphate which is facing now the cytoplasmic side of ER. Now the remaining 4 mannose residues attached with GDP, we transfer them to the Dolichol phosphate carrier, and from here we have this. Okay, now we want them in the luminal phase of ER. So what we do, we flip again. Now this would face the luminal phase of the ER. Here we have. Okay, we transfer it to the already formed this much chain of the oligosaccharide. Here we have 4 more residues of mannose. After this, the carrier which was containing 4 residues, the Dolichol phosphate, it is flipped back to complete another round of the transfer of these sugars which are present in the cytoplasm. Next is the turn of glucose residues. They are present in cytoplasm. We will repeat this step. 3 glucose residues are there which are attached to the UDP. Okay, they are transferred to the Dolichol phosphate and we flip this, and this is here, and we give it back to the already growing chain of oligosaccharide, and we are finishing here. This triangular red color is showing the glucose molecules. So this is the completely formed oligosaccharide chain. Now it is the turn to transfer this completely formed chain to the protein, and we are talking here about N-linked glycosylation which is happening in ER. Okay, when it is transferred to the protein which is now into the ER lumen, okay, what happens next? 3 glucose residues which we added, they are removed by an enzyme which is known as glycosidase, and one particular mannose residue is also removed, and after this one glucose residue is added to complete the modification of this oligosaccharide chain in ER. And after this chain is ready to sent in Golgi. So we can say N-linked glycosylation act as a base for further modification of this oligosaccharide chain in Golgi. So this was all about the glycosylation process, and if you liked my efforts, please subscribe to my channel. Thank you.